speaker is Dr. James Liao, Chancellor's Professor of Chemical and Biomolecular Engineering at UCLA. Dr. Liao received his BS degree from National Taiwan University and his PhD from the University of Wisconsin-Madison. After working as a research scientist at Eastman Kodak Company in Rochester, New York, he started his academic career at Texas A&M University in 1990 and moved to UCLA in 1997. In the last few years, Liao has received widespread attention for his work in development of biofuel production, for the production of more efficient biofuels by genetically modifying E. coli bacterium, and most recently for modifying Cyanobacteria to consume CO2 to produce isobutanol. Among his many honors, he received this year's Presidential Green Chemistry Challenge Award from the U.S. Environmental Protection Agency for his groundbreaking work in recycling carbon dioxide for the biosynthesis of higher alcohols. The title of today's talk is Biological Synthesis of C3 to C8 Fuels and Chemicals.
Well, thank you, Kelly. Very much appreciate this invitation. I had a very unique experience today by talking to many postdocs and grad students. Usually you go to this seminar talks, you all talk to the faculty member. It's very rare to talk to a real working power. So it's really a pleasure to talk to grad students and postdoc in EBI. So I'm going to talk to you about some part of our work. And my talk is divided into two parts. One part, the first part is more on the synthetic side. The second part is more on the analytical side. Okay, the first part, synthetic side, probably many of you, if you go to meetings, you probably have heard me talk about that. So I will be a little bit fast, because quite a bit of information there, and most of them I've presented and published. The second part, analytical side, is relatively new, and I don't remember I've talked in public meetings as much, at least maybe once or twice. So I will probably spend more time here. So biomass is not new to this group. So I just have to go to this slide to normalize what we are talking about. We try to recycle CO2, eventually go to fuel. And this cycle, we first take to biomass, and then break down lignocellulose to sugar, and then take sugar to biofuel. In this particular case, is ethanol. So now if we look at this cycle, we have 1 carbon in CO2. We make into thousands of molecules in the lignocellulosic materials, and break down to 6 carbon or 5 carbon sugar, only to break down to 2 carbon. Okay, so this is the cycle we are currently in. It doesn't really make a lot of efficiency argument here, except that making the biomass is extremely efficient. Human beings have been working on this for thousands of years. This is primarily the reason that we are using this kind of scheme. But if we have our way, we would like to cut the intermediate point, directly synthesize fuel, whatever that you like, from CO2. So now we can directly take CO2, 1 carbon, and build whatever number of carbon of fuel that you are interested in, if we can make that efficient enough. Of course, the idea is simple, but make that efficient is not that easy. So that's why we haven't seen this in reality yet. So to summarize the biofuel problems, I'm sure people in this room are very familiar with this. Is the overall production efficiency is not high enough. Efficiency is not a major problem if you are willing to pay the money. So you can make something very efficient by spending a lot of money. So that's a catch-22. You want to increase the efficiency, you want to remain the low cost. That's the difficult part. In order to remain the low cost, you need to make sure you have a high throughput or high flux production. Okay, making a molecule and detect that in GC mass spec is already very challenging. I'm sure most of you experienced that. You see the first peak, you are very excited. But that's, of course, is not going to solve the whole world's energy problem. You need to make that in tons and tons of quantity in the foolproof manner. So the problem summarized here. Another implicit problem is that up until very recent years, the biofuel considered very limited, primarily ethanol and fatty acid esters. So those are the, this is biodiesel, of course. So those are the typical biofuels that have been considered in the past several decades until very recent. So we and others have been thinking about, can new fuels if we synthesize new fuels, can we change this landscape? Can we improve the throughput of production? Can we reduce the cost? Can we improve the overall production efficiency? This is not very obvious because most people will say, no matter what you do, you have to cut down, you have to degrade the lignin cellulose, you have to degrade to sugar, and so it doesn't matter, once you have sugar, you can go whatever you like. So changing fuel production may or may not improve any of these factors that you're interested in. Well, it may not be obvious, but perhaps you'll find some surprises. So the challenge of scientists and engineers is to create new possibilities, new varieties, so something new might happen. So that's the basis that we went into this field in the beginning. So we like to eventually reach this goal by direct recycle CO2 to biofuels. And to do that, we'll use the photosynthetic machinery that takes photoenergy to split water, and in the process produce ATP and reducing power. And this ATP and reducing power will then fuel the carbon dioxide fixation cycle. And I intentionally put this on because this cycle was identified by the namesake of this building. So I feel that it's very appropriate to show this slide particularly. And the CO2 incorporated into Kelvin cycle primarily for the native organisms is to build the sugar. Okay? Now, if we can somehow divert the flux to make whatever fuel we're interested in, we'll be achieving something new and may eventually become viable in a large scale. So what kind of higher alcohol we're interested in? Okay, we are interested in anything longer than 2 carbons. So that includes 3 carbon, 4, 5, 6, 7, so on and so forth. And once you have more than 2 carbons, you begin to have branching patterns. So we're interested in those. Those may have different chemical properties, but as a fuel, they're pretty much similar with some minor exceptions. Sometimes for internal combustion engines, some branched chain fuels may be better because of the higher octane numbers. But for other applications, maybe not as good. So among all these higher alcohols, only 2 of them have been produced biologically in large scale. One is 1-butanol. This is produced by Clostridium. The other one is isopropanol. It's also produced by Clostridium species. The rest of the longer chain alcohols have never been produced by microbial processes. They may have appeared in literature from time to time, but primarily from amino acid degradation byproducts. And particularly if you go to longer chain and much more complicated structures, then it's hard to prove that was actually a result of microbial reactions. So when we start this project, we look at how microorganisms produce alcohols in general. Okay, start from the simplest pathway, ethanol. This is the most ubiquitous ethanol production pathway. It exists in E. coli. It also exists in human beings. And human beings, of course, reverse this reaction so that we can enjoy ethanol. Now, this start from pyruvate, which is the degradation product of sugar. It's a keto acid. First go through the oxidative, reductive oxidation. And then to 2 more steps of reduction, it go to ethanol. Now, this process has been extended by Clostridium to make longer chain alcohol, butanol. As you can see, this process, for those of you who work on this, you immediately recognize that this process is identical to the fatty acid synthesis pathway, except that instead of using ACP, you use CoA, coenzyme A, to activate the reaction. Okay, now you see every intermediate has CoA. Okay, why do we have all this so much CoA stuff in there? Well, for a very simple reason. This kind of reactions are difficult to achieve, and biology solved this problem by activating the compound using coenzyme A or ACP. So this part of the pathway has been successful in nature only in Clostridium, and probably that's the only known organism, or known type of organism that synthesizes N-butanol or 1-butanol. And apart from that, not very many examples exist. So when we start working on this, we were trying to move this pathway into E. coli. Well, we achieved 1-butanol production. And but we were not able to improve that very much. So at that point, we decided to look for alternative pathways. Starting from ethanol, we know that there is a very common pathway that bypasses coenzyme A that is directly going to decarboxylation without harvesting the NADH from pyruvate. So this process is utilized by Saccharomyces cerevisiae and also Xymomonas mobilis. For those of you who work in this area, which is about 99.9% of people in this room, you know that these two organisms are the only two organisms that are used industrially to produce ethanol. So immediately you get a hint that this process, this pathway is much more efficient than this CoA intermediate pathway. So now our challenge is, can we extend this pathway to longer chain alcohols? Okay. Well, nature hasn't done that, but can we, as an engineer, extend this pathway to make longer chain alcohol? That becomes our challenge. To do that, we start the chemistry of keto acid decarboxylation. This is the keto acid going through the decarboxylase, become aldehyde, and then ethanol. Now, if we want to use a longer chain keto acid, what we have to do is to find an enzyme that can conduct the same type of reaction, but instead of pyruvate, now we stick an R group at the end of the methyl group, okay? So to make longer chain alcohol. So if we can do this, we can make longer chain alcohol. Now this pathway, this process has been described almost 100 years ago by a German group, but was never explored further. So how do we do that? Fortunately, these days, the crystal structure have been available. So this is Xymomonas pyruvate decarboxylase. The active site is depicted here. These are the residues that are known to interact with the substrate. And this is the TPP cofactor. Now, if we want to stick a larger substrate in here, we have to either enlarge this cavity or just go into nature, find something that already exists. Okay, fortunately, we found something that's Lactococcus lactis, keto acid decarboxylase. This has the same type of reaction, same type of structure, but with a larger substrate, the active site cavity. So we clone this enzyme into E. coli along with the appropriate alcohol dehydrogenase. Alcohol dehydrogenase turned out not to be a major problem. There are many examples, many species contain different kinds of alcohol dehydrogenase you can use. So we cloned that into E. coli, and we fed the E. coli with various keto acids, and sure enough, we detected different kinds of alcohol. So that proves that this kind of chemistry works well in E. coli. So now we solve half of the problem. The longer chain keto acid can go to longer chain alcohol. The more difficult problem is how do we extend the chain length from pyruvate to longer chain keto acids. This is one of the shortest chain lengths of keto acids, 3 carbon, and then how do we extend that? It turns out that the carbon-carbon bond chain elongation is one of the most difficult problems in biology. And I can only find 2 examples. Okay, one is fatty acid chain elongation. The other is isoprenoids, and nothing else. Okay, all other chain elongation pathway involve either CO bound or CC or CN bound. I cannot find any other CC bound iterative chain elongation. Maybe do it once, but not iterative fashion. So fatty acid chain elongation, this is well studied. We start from acetyl-CoA, go to malonyl-CoA, and then go through a series of reactions. Okay, the key point is that at the end of the reaction, it increase the chain length by 2. So this is what we call the +2 fatty acid chain elongation pathway. If we take the essence of this pathway, we quickly find out that the essence can be represented in this way. You start from a substrate that has a certain chemical group, functional group. You go through a series of reactions. For those of us who are not that excited about chemistry, we just represent with A, B, C, D. At the end of the chain, you get the original functional group back, but with the chain length increased by 1 unit. And that 1 unit could be 1 carbon, 2 carbon, 3 carbon, 4 carbon, or 5 carbon. So with this in mind, all we have to do is to find a set of pathway that can conduct this type of chemistry. Okay, starting from a known functional group, go through a series of reactions, retain the same functional group, but increase the chain length by 1 unit. So there are a couple examples exist in nature already. One is called acetyl hydroxy acid synthase. This is the enzyme catalyzes the first step. Okay, again, you see that this is the keto acid. After a series of reactions, let's not worry about the details. At the end of the day, we get the keto acid back with the chain length increased by 2 with a branch. This pathway is used in valine synthesis in a non-iterative fashion. So immediately we recognize that this pathway can be used to increase the chain length by 2. Starting from pyruvate, now you can go to this keto valerate. And then you can use that to derive alcohol. Okay. If you're clever, you can go back to do this more iteration. We'll talk about that in a little bit. So with that in mind, we immediately can expand this decarboxylation pathway to various kinds of alcohols by using this type of chain elongation. So in particular, from pyruvate to ketoisovalerate, we can add 3 more groups in the valine biosynthesis pathway, immediately increase the chain length by 2, and another decarboxylation, we become a 4 carbon. This is a 3 carbon, increase to 5 carbon, get to 4 carbon. And the steps involved are very analogous to the ethanol synthesis. Ethanol starts from pyruvate and go to ethanol with 2-step decarboxylation. All we have to do is add 3 more steps, increase the chain length by 2 with the branch. And then we can go to the decarboxylation. Again, we get alcohol back. The theoretical yield for ethanol production from glucose is about 51% on the weight basis. For isobutanol, it's a little bit less, for 41% on the weight basis. Because isobutanol contains a little bit more energy and has a little bit better fuel property, so it offsets the inefficiency in mass yield. So to do that, we clone various genes and shunt the pathway off from valine biosynthesis and make that isobutanol. And I'm not going to go to detail. I don't want to make it sound too trivial. Okay, because my postdoc, my grad student did spend a lot of time to making it work. I'm going to skip to the final result. So after the various ways to optimize this, and by the way, everything is published, so you can go to the literature to find out how we actually finish this. So the bottom line, the first round of optimization gave us about 20 grams per liter of isobutanol in about 100 hours. Okay, and the yield is about 86% of the theoretical maximum. So how does that compare to the benchmark? The benchmark we use is the cross-reactant production of 1-butanol, which typically achieve a little bit less than 20 grams per liter. This is probably the best in the literature. And typically in companies, there'll be about 15 grams per liter. You have to push it very hard to get to that level. Of course, I'm talking about batch reaction. I'm not talking about the more advanced type of bioreactors. And the yield is about 80%, a little bit more than 80%, so very comparable to what we have done. Okay. And a little bit of optimization, we now can push the isobutanol in bioreactors to 40 grams per liter. So you know this is doable. Okay, the whole pathway is working very well. There's no major problems. It almost works as well as we like. So that was the elongation, chain elongation using AHAS pathway. That's at 2 carbons. There's another pathway that you can use, another mechanism you can use to go to the chain elongation, and that is depicted here. It's called the isopropyl malate synthase pathway. Okay, this is isopropyl malate. The first enzyme is isopropyl malate synthase, or IPMS. Again, we see that for a series of reactions, you start from keto acid. After a series of reactions, you get back the keto acid with the chain length increased by 1. Okay? And this is used in leucine biosynthesis. Again, nature never evolved this for more rounds, but at least we can use it for the chain elongation. Now we have 2 ways to do chain elongation. Okay, one is adding 2 carbons, the other one add 1 carbon. One with the branch pattern, the other one doesn't have branch. And then we can use that in a combinatorial fashion. Okay, we can get to various type of alcohols. Okay, for example, after this isoketo valerate, we add another +1 chemistry, we get to 3-methyl 1-butanol, the 5-carbon compound. Okay. Or we can directly shoot off from threonine, the degradation pathway, go to ketobutyrate. This is 1 carbon longer than pyruvate, and directly decarboxylate that to make 1 propanol. This is also very simple. We can also take this as a raw material, as an intermediate, and go through the ILV pathway, and then to make that 2-MB. Alternatively, we can make 1 butanol, which we were set out to do initially. Now this is the LU-ABCD pathway. That's an IPMS pathway that's commonly exists in all organisms, but it's used for this leucine biosynthesis and not used for this. And this branch pathway actually is a byproduct only exists in high throughput. And this has been a problem for protein production because the natural outcome of this is the production of a valine analog called norvaline, which got misincorporated into proteins and caused a lot of problem. Okay, now we can take advantage of this kind of property. Instead of making it into norvaline, we can now make that to 1-butanol. Also very convenient. And it's shown here. So this is the typical pathway for leucine to biosynthesis. The precursor for leucine is this keto acid called ketoisocaparate. Okay. We want to use the same type of enzyme, same type of chemistry, but smaller substrate this time, without this branch. So we feed ketobutyrate, we can get to this. Okay, this is how we can do 1-butanol. So instead of the CoA-dependent pathway, now you build yourself a complete different set of pathway that's independent of coenzyme A. So it may have some advantage over the others. So we have demonstrated we can synthesize these keto acid derived compounds from sugar. Now, our ultimate goal is to directly recycle CO2 to these fuels without going to the sugar intermediate. So we decided to insert this pathway into Synechococcus elongatus, which is one of the most commonly studied cyanobacteria. The genetic system is very well worked out. So we integrate the genes that we talk about from pyruvate to isobutanol. And sure enough, we did see isobutanol coming off the fermenter, coming off the flask. Although this is relatively low, but this is definitely an encouraging result that you can directly synthesize isobutanol from CO2. And if we do a little bit of small-scale engineering, we bubble CO2 in this flask. And we see that all the product, in this case, we use isobutyraldehyde because it is more volatile, coming off the fermenter, coming off the flask. And the fermenter, the flask, maintained a very low concentration of isobutyraldehyde, which is extremely good to us because this type of compounds are toxic to the cell. So now you achieve simultaneous production and product removal. And therefore, you can continuously produce this compound for more than 10 days without loss of productivity. Okay? Although we haven't pushed it further, but if we can push it further for several months, then we begin to talk about continuous production of this compound, which is very attractive. And the OD stay constant. OD is not changing. One interesting thing in this project is that we actually integrated the Rubisco into the system as well. As we know that Rubisco is the most abundant protein in the world. And many people are trying to increase the photosynthesis rate by increasing Rubisco copy number without any effect, because probably through billions of years of evolution, the organisms have already optimized Rubisco. But one thing different here is that we are not asking cells to grow faster. We are asking cells to produce more. So instead of this increase the flux to growth, which is very difficult, now we shunt it to isobutyraldehyde or isobutanol. We see that overexpression of Rubisco just a little bit, not too much. Okay, we can actually increase the production about 20% more. And instead of going to a little bit less than 1 gram, now we can produce more than the 1 gram. It's about the 1.2 gram in about 8 days. So this is also an encouraging result showing that the capability of cyanobacteria has not been fully explored, and there's a there's a good chance that we can do a lot better. So how does our cyanobacteria work compared to the existing platform of ethanol production? So this is the independent review by John Sheehan, who used our data here. This is our data from isobutanol directly from CO2, and this is isobutyl aldehyde from CO2. Of course, this is only flat scale, not to compare with this is the real production scale. But it looks very interesting. The current production from corn ethanol, the productivity per acre, per hectare, is a little bit less than 4,000. And the current cellulosic ethanol, I don't know how accurate these numbers are, but this is what we get from this review. So our data compare very favorably, and also pretty promising compared to the current algolipid processes. So you can see that even though we have not demonstrated a large-scale production, but the initial result, this is almost in stage 0. It's quite promising. So this is our ultimate scheme of production to using, of course, we're not going to use this as an ultimate scheme, but from CO2 to biofuel in one shot instead of going through the intermediates. It's still a long way to go, but the result is encouraging. So up until now, we talk about production from pyruvate to various alcohols, C3, C4, C5, with different branch pattern. Now we can push this further. Okay, we talk about this kind of chain elongation pathway that used in leucine, valine biosynthesis. Now if we can engineer the proteins so that it can iteratively cycle through this pathway, we can increase the chain length more and more. Okay, so that's what we did. So we use this IPMS protein, or LUA in E. coli. We engineered this pathway, and fortunately, instead of engineering the whole pathway, we found that only 2 enzymes we need to engineer. One is the LUA, which determines the chain length primarily. The other one is the decarboxylation pathway, is this pathway, this enzyme. So you engineer these 2 enzymes that were sufficient for us to modulate the chain length. So in this case, we have to do some rational design by looking at the binding site. Okay, this is my former postdoc, Kachin's work, who is now assistant professor in Minnesota. So Kachin did was looking at this protein structure and identify the essential, whatever the likely residues that restrict the active site volume, and engineer those residues to increase the active sites and see how that works. It turns out that Kachin was able to iteratively synthesize these different chain lengths or pathways. So the pathway initially did only one pass. Okay, now we're able to cycle through this pathway 5 times, making to a heptanol, okay, the octanol even. And different mutations have different productivity, different yield to different kind of chain lengths. From butanol, pentanol, hexanol, heptanol. We're able to make the longest chain length was 8 carbons. And even this type of strategy can be used to make aromatic alcohols, phenylpropanol. Propanol has never been demonstrated to produce biologically. So this shows that this type of synthetic iterative chain elongation pathway has a potential and has never been explored. So this type of strategy allows us to produce a variety of alcohols. This box shown in pink are the ones that go through multiple rounds of chain elongation. So what are the advantages of this keto pathway? One is it's very ubiquitous. So almost all organisms contain amino acid synthesis pathway. So you can presumably move this pathway into any organism you like, and you have a good chance to be successful. And this pathway is very pliant. You can engineer that to the extent you like. Finally, it has a built-in driving force. This has never been appreciated. Now let me explain a little bit what is the driving force I'm talking about. These are the different pathways making ethanol, isobutanol, 1-propanol, and 1-butanol. Among all these things, ethanol produced very well. Everybody knows that you can produce ethanol in a huge quantity. Isobutanol, we've shown that we can produce that very easily. Isopropanol in our other work, we show that it can easily produce as well. Now, very similar pathway to isopropanol was 1-butanol. Okay. Almost identical pathway with something different. But our group, including our group and other groups, have tremendous difficulty in moving this pathway to a non-native organisms. So we ask the question, why? Why some pathway you can move to different organisms, others you cannot? If you look at these pathways, okay, immediately you see the differences here. This pathway, at the end, evolves CO2. This pathway, many places, at 2 places evolve CO2. And this pathway, also at the end, is CO2. And this pathway has none. So it appears that CO2 evolution becomes a driving force. So this is an entropic driving force. Once CO2 is evolved, it goes off, gets diluted. So we almost can hypothesize that there's a driving force, or pulling force, if you will, that pull the pathway down to ethanol very easily, pull the pathway down to isobutanol very easily without competing with, and pulling against other competing pathway. The same is not true for 1-butanol. Okay, this driving force is too high, so acetyl-CoA goes to somewhere else. So that may be a reason why it didn't produce so well, and that may be a reason why isobutanol is so easy to produce. And isobutanol is useful as a fuel. It can also be hydrated to make isobutene and make into various kind of chemicals. So it is not only a potential fuel, but also a good chemical feedstock. In addition to making alcohol as a fuel, the same strategy you can make non-natural amino acids. Okay, so we have done this as an example. This ketobutyrate is a degradation product of threonine. And you can go through the 1 round of amination, you get to homoalanine. Okay, there's one more chain compared to alanine. This is a non-natural amino acid, very useful in pharmaceutical intermediate, very useful as a pharmaceutical intermediates. Immediately, we can find is that it's a key intermediate for 3 major drugs. This is ethambutol, is a first-line TB drug. It's a more than $1 billion market. This is levetiracetam. This is trade name Keppra. It's also a $1 billion drug for epilepsy. This is the second line. This is the next generation drug for this Keppra. It's on the clinical trial stage 3. All combined, there are more than several $1 billion of product that you can make out of homoalanine. So this is, again, another variation of the same thing. You can build the keto acid pathway and then shoot it off to non-natural alcohol or shoot it off to non-natural amino acids. OK, so the rest of the time, I'm going to spend more on the other side of my work, more analytical. So far, we're synthesizing stuff. OK, now we want to analyze how we do it so well and what are the problems and how do we solve them. One of the common problems I'm sure everybody knows, and we are dealing with it, is the tolerance problem. OK, what do I mean by tolerance? Everybody knows that isobutanol, butanol, and those high alcohols are toxic to the cell. This is the Y-type cell. Without any isobutanol, it grows just normally. If you put in 6 grams, it inhibits growth severely. 8 grams is almost no growth. 10 grams, it starts degrading. It starts disrupting the cell. 15 grams is almost dead completely. So the common thought is that toxicity is a major problem. And if we define tolerance as the growth rate under the given concentration of product, so that implies that this definition argues that tolerance increase, growth rate increase. So intuitively, everybody will say, hey, this is good, tolerance is good, growth rate is good. These are all good attributes to production. But a few years ago, we actually derived an equation. This was published finally last year. We showed that growth rate is proportional to the square root of biomass yield. Biomass yield is defined as the cell mass formed divided by substrate consumed. Now, because of mass balance, biomass yield is equal to 1 minus product yield. So now you can see growth rate increase, the product yield will decrease. So based on this argument, you almost argue that increased tolerance, you will decrease the product formation rate. Of course, this is a theoretical argument. We don't know whether it's true or not. So we went on to actually, well, first we proved this relationship actually is true or not. So we grew E. coli in various nutritional concentrations. This is varied amino acid concentration. We measure the growth rate, normalized to a common standard, and measure the biomass yield. Sure enough, we found a straight line correlation between the growth rate and the square root of biomass yield. If you grow E. coli in different carbon sources, glucose, mannitol, arabinose, and so on and so forth, again, you see this square root relationship. This is assuming that this is the equation we derive, and we attribute to this result to the fact that these two factors are not changing. Kcat is the Kcat of the limiting enzyme, whatever that is. Rm is the total protein synthesis rate. And so we assume that these two are constant, and that will explain this result. Now, there are conditions where you can alter these two properties. And for example, if you grow the cells in different concentrations of ethanol, you see growth rate decrease. You measure Kcat, you measure yield, growth, biomass yield, you measure Rm, they all change. But if you plug into our equation, they all come back to the same straight line. Okay, mu is equal to square root of y, the biomass yield times Rm times Kcat. So that will argue that if you increase the growth rate, you'll decrease product yield. So because product yield and biomass yield is just related by mass balance. So 1 over product yield is equal to the biomass yield. So to test whether this argument is true or not true, tolerance may or may not increase production. We actually select a mutant E. coli that is more tolerant to isobutanol. So what we did was we conducted a series dilution of culture from E. coli, and with increasing concentration from 4 grams per liter to 6 grams to 8 grams. And then at the end of the transfer, after 45 transfers, we isolate the single colony and test the tolerance. So this is the wild type. It decreased the growth rate by different concentration of isobutanol. For our mutant, you see it was able to grow in the presence of 6 grams per liter or a little bit of 8 grams per liter. So they are definitely more tolerant to isobutanol. So then the first question we ask is, what's happening in these cells? How do they become a little bit more tolerant to isobutanol? So we sequence the whole genome. The whole genome is sequenced, and it contains 25 insertion sequence elements. They insert in various positions, okay, practically knock out many genes, and it also includes a large deletion fragment, okay, between these two genes in this position. There's only one SNP in DNA polymerase, and probably when we demonstrate that doesn't have any effect. So how do we know among all these mutations which one are important? So all the single mutations, we repair them from this tolerant strain. This is the wild type. It doesn't tolerate the isobutanol concentration of 8 grams per liter. This is the mutant. It tolerates the 8 grams per liter of isobutanol. So we repair each of these mutations and try to see whether the tolerance decrease or not. So most of them decrease a little bit. A few of them decrease dramatically. So we call these essential mutations. Okay, these are 5 essential mutations that give rise to this property. So based on these 5 essential mutations, we can reconstruct this tolerant strain. And then we can test how tolerant they are. So these are the 5 mutations that we call essential. We reintroduce them into wild-type strain, trying to reconstruct that. If you introduce each one individually, the tolerance increases just a little bit. This is the mutant we get. All the single mutation introduced only improve a little bit. Double mutation increase a little bit more. Now, only when we introduce all 5 of them were we able to reconstruct almost the identical behavior of our original mutant. That indicates that these 5 are necessary and sufficient to reproduce this similar phenotype. Okay. So now we have a mutant that's resistant to to isobutanol a little bit more than wild type, and we know what are the essential mutations. There are 5 of them, and each one we can talk about their function. Okay. One of them particularly interesting is this YHBJ gene, which is a predicted protein containing ATPase. And others are, for example, the ACRA, which is surprisingly, this is the efflux pump. Now, it's not the overexpression of this pump, it's the knockout of this pump that caused the tolerance. And similarly, the MAR gene, this is also a multi-antibiotic resistant gene. Again, knocking out of this pump helped to tolerate isobutanol. This is a surprise to us. So what does this YHBJ gene do? It actually caused the enhanced degradation of this small RNA called GLIM-Z. This small RNA increased the expression of GLIM-S, which is the enzyme that synthesizes an intermediate lead to peptidoglycan synthesis. So it's an important compound in membrane. So to test the hypothesis that this peptidoglycan is actually involved in this tolerance, okay, we actually overexpressed GLIM-Z. So we overexpressed GLIM-Z, we're able to reconstruct this behavior of tolerance in the YHBJ mutant. Similarly, if we say this peptidoglycan synthesis is important, we should be able to supplement the culture with glucosamine, okay, because that's another feed point into this intermediate. And sure enough, if we fed this culture with glucosamine, we're able to increase the tolerance to a similar level as our mutant. So that indicates that this peptidoglycan is also important in this tolerant strain. So now we have a tolerant strain, we know the essential mutations, we know potential mechanism, not completely, but we know partial mechanism at least. We finally are ready to test whether it actually helps the production. So we grew these cells in culture with the same condition to produce isobutanol. To our surprise, they produce identical amount. Okay? All come out to about 20 grams per liter, whether they are tolerant or not. The only time they perform better is when we initially give the culture with 8 grams per liter of isobutanol. In that case, wild type could not even grow. So they stay there, they produce nothing. The mutant could continue to grow and produce isobutanol. This is the only chance that it will win. And this is somewhat artificial. I mean, I cannot think of a reason that would be practical in this case. So if you look back in our initial production curve, you see most of the isobutanol was produced when the cells are not growing. So this is another reason why improved tolerance is not directly related to product formation, because most of the cells are already dead. So we're essentially turning a cell into a biocatalyst without any biomass formation. You also argue that this type of reasoning may work, although we are not utilizing the growth part. But if you are growing and produce, of course, you're going to waste the carbon source. So that's a story about our square root relationship story. Now, remaining the few minutes, I'm going to talk about model-aided strain improvement. So one of the difficulty in this business, I'm sure many of you will recognize, is that you do a lot of work in constructing the pathway. Now you defend in front of your committee. You can't really say too much because many of the approach you use are trial and error. Okay, there are not too many exciting things you can brag about. That's good and bad. The good thing is that there are a lot of new technology, new discovery remain to be made. The bad thing is that it's hard to make those discoveries. So with that in mind, we try to develop new tools, particularly new mathematical tools to help us to decide what to do to improve your production. And this is depicted in here. Typical cycle of strain improvement. You start from something, utilize current knowledge, and then you generate intuitive targets for deletion or overexpression. You conduct those experiments in the lab, and then you do fermentation. Then you decide whether, okay, I have overexpressed enzyme 1, it decreased the production a little bit. Overexpressed enzyme 2 increased twofold. And that's almost at the end. You can never be able to go back to this cycle and utilize this information to the next, at least not in a systematic way. Okay? So this loop is now discontinued. So you have to, again, rely on your intuition, your intuitive reasoning. So the challenge becomes, how do we construct a model utilizing this type of data? The current way of modeling metabolism, probably the most common way, are two. One is the stoichiometry base. You put your metabolism into this stoichiometry, and whatever knockout, you knock out this column. Okay, this is a common way to do that. The problem of this type of approach is that if you have an alteration that changes Vmax, for example, you overexpress a gene or change a Km, then there's nothing you can change. So this approach is inadequate for that. So the alternative to that is the traditional kinetic model, that you put in an ODE model, you get the equation, and then you try to get the parameters from literature, or you measure that. And the problem there is that there's just too many parameters. And even though you have parameters, it is not transferable to the next organism you're working on. So this is not practical. So what we come up with is another approach. We are saying that you don't have the kinetic parameters, and you don't have the dynamic data. Nobody will measure metabolite concentration profile for you. I will be surprised if EBI spend a lot of effort trying to measure metabolite concentration as a function of time. You may have occasionally, you make 1 or 2 measurements, but this is certainly not everyday life. What we commonly do are this type of data. You overexpress the gene, find out how much product concentration increase or decrease. You knock out something, you find out whether it increase your production flux or decrease your production flux or no effect. Those are the type of data you have every day. How do we use that data in the modeling? We first recognize that those data basically are the steady-state flux. So we do know steady-state flux, even though they are not very accurate, but we know roughly where they are. So if we can construct a set of models that converge to the same steady-state flux that you know of, at the same time allow it to assume any kinetics that are allowable by thermodynamics, then I call this an ensemble of models that describe appropriately the cell you begin with. Of course, you know, so far we utilize only the steady-state information. Our kinetics can take anything we want. So some of them are accurate, most of them are junk. But at least you now have a way to shrink the possibility to a very small number. Among all these models, how do you know which one are appropriate? Then you do a perturbation. This is exactly the same way you do experiment. You take this cell, you overexpress the enzyme, and you watch whether the production flux increase or decrease. You overexpress the same enzyme in your model, and then conduct a simulation. Now, this time, it will not reach the same steady state. It go everywhere. Some increase, some decrease. You retain the ones that are consistent with your data. For example, you overexpress this particular enzyme, the flux range around here. You retain those models that behave the same. Discard the rest. Then you enrich the models that are accurate, at least approximately accurate. And then you can cycle this. Go on to the next enzyme, overexpress that, or downregulate that, or knock it out. Whatever you like to do, you do the same on your ensemble, compare your result, you enrich the model. A few more times, you begin to converge. Okay? It sounds simple, but actually it's not that simple. Okay? But the idea is this. So now you have a way to start from an ensemble of models that takes any kinetics. Okay? You enrich the correct kinetics by perturbing the cell the same way the experimentalists perturb their cells in the lab. Overexpress an enzyme, downregulate an enzyme or knock out an enzyme. And you measure exactly the same thing that experimentalists are measuring every day. How does butanol production increase? How does glucose consumption increase? That's the same kind of data that you get from your neighbors in the lab. You use that in the screening of a model. Discard those models that don't describe the same way the data do. And then you can enrich the model. Using this way, even though we have a lot of parameters, but because we constrain the parameters to the same steady state, that essentially shrink the parameter space to a very small area. And that is the reason why our model can work. Otherwise, if you don't do that, you just start from all the kinetic parameters without constraining the steady state, you will sample a huge space. And for those of you who did any modeling effort, you know that constructing model is difficult only because you cannot get a steady state. Most of the parameters you get, you don't get a steady state. If you start from a set of models that reach a steady state, then you can play with very efficiently. So this tool allows you to zoom in the models that are potentially fruitful, and you can screen among that. This is an example. We start from about 4,000 models, okay, not too many, and then go through several screening. So for example, this is the deletion of SDH gene. Okay, this is the succinate production. It changed the flux a little bit. Okay, we compare the model with the data. We retain about 2,000 that are consistent with this prediction, this data, and then knock out POX-B. The model number dropped to 800 or so. Another one dropped to 200. Another one dropped to 200. The more we get, the more we converge. In this particular case, it almost stabilized. In more of our case, after 4 or 5 iterations, the number of models stayed constant. That means the same type of models continue to predict the correct answers. So we begin to have something that can describe your system appropriately. Then you can use that to predict what might happen if you knock out this gene, if you overexpress that gene. So this is the idea. Of course, we can use a lot of literature data and show that we are so smart we can do that, but nobody will believe that. So we use actually a real case. This is the production of 2-methylbutanol. This is one of the things that we're interested in. As you can see, we showed that earlier in our slides. And there are 2 ways to do that. One is go through the threonine pathway to produce 2-Kb. Another way, which I haven't shown, which is published, by the way, already, is build an alternative pathway to get to 2-Kb. That's through this SIM-A pathway. This is much shorter than that. So the student Anthony did this, okay, along with Claire and Mike and others, grad students. They did a lot of work on this. They knock out genes, overexpress this, overexpress that, practically do everything they could. Generate lots of data, shown here, some increase, some decrease, some with no effect, and so on and so forth. And get to a point where they don't know what to do next. Okay. All the tools are exhausted, nowhere to do. So now it's time for this modeling person come in to utilize all the data to screen the model. Okay, this is coming the right time where we develop this modeling tool. And putting all this data, we screen all the ensemble. We found out that if you overexpress this gene, PTA, the effect is very small, effect on the 2MB flux. Overexpress that, very small, so on and so forth. Some of them negative effect. Until here, you overexpress IRV-GM, boom, it stands out. So this model prediction suggests that overexpressed IRV-GM is the most effective one to improve the production strength after that. And sure enough, and this one is not easy to predict because this is a linear pathway. It's not a branching pathway. This is here. How do you know it's this? It's not here. Okay, what do you know is not this pathway or that one. We struggle a lot with that. And with this piece of information, we're able to overexpress this, even though we already have overexpressed the plasmid in the cell. But apparently it's not enough. So we need to continue to overexpress that. And after that's done, we're able to increase the 2MB production up to about 10 grams per liter. Previously I think was half of that. So we're able to increase the production by about 50 to 100%. By the way, this 2MB production is considered quite difficult because this one is more toxic than isobutanol. So even though we can produce 20 gram or 40 grams of isobutanol, 2MB has remained relatively low. So this is an example of using this theoretical tool, modeling tool, to help experimentalists decide what target to use. Maybe we're lucky this time. We haven't get to an extensive test of this tool yet, but at least the initial results seems interesting. Of course, it's not to say that we have no incorrect prediction. Believe me, we have a lot of incorrect predictions. We are working on that as well. So to summarize what we said, pretty much we focus on the keto acid pathway, and we show that they are highly effective, transferable, and extendable. And this keto acid pathway can be the basis for synthetic iterative chain elongation. We can completely iterate this one for several rounds and go to the aldehyde keto acid. We then can make the non-natural alcohols or non-natural amino acids. And then we show you that we have a few design models that help to interpret data and gain new insights. And hope this work will stimulate your further thoughts and to make this area much more productive. And I thank you for your attention and be happy to entertain any questions. Before I close, I have to acknowledge several people here. This is my group, probably already a year out of date. Many people already left. Two key people, Shota Atsumi and Kachun, are not shown here. They already left, unfortunately. But those are my group. And my funding comes from NIH, DOE, NSF, particularly UCLA, DOE, Institute for Genomics and Proteomics. Thank you.
I wanted to ask you, you cited 20 grams of isobutanol produced over a period of time, and I wanted to make sure how much biomass are you generating at the same time? Sounded like no biomass.
Not very much, yeah. So this is OD. OD is about, what, 6, 7? So relatively low. It's 20 grams per liter, the 6, 7, and stay there for a long time.
That's for a period of 5 days.
Yeah,
right?
yeah.
How long can you maintain the cells in this?
Well, usually 3-4 days they start decaying. This is a good run that can last for 5 days. But unless you do a very careful job, they usually start decaying after 3 days.
So this is with with E. coli.
This is E. coli. This is E. coli.
Glucose as a substrate.
That's correct. Yes.
You found that the yield for both the tolerant strain and the non-tolerant strain is almost the same?
Yes.
So
yes.
How do you think that's going to impact bioprocesses where they're trying to engineer cells for toxicity? Do you think?
I don't know. I think we don't have a definite conclusion yet. At least we show you an example where the cells are tolerant, but they don't produce more. I think similar results have been found in Clostridium as well. Actually, the opposite result. In those cases, people evolve Clostridium strain that are more tolerant to 1-butanol and show that produce even less. But that may be because at that time people are attributing to the deleterious mutation accumulated over the time. But in our case, I don't know what's going on. At least it's not always true that increased tolerance lead to increased production. At least we can say that. Yes.
So, I mean, to me it seems really kind of clear or obvious that at a certain threshold of isobutanol, other things are just impacted so that essentially growth is completely stopped. If some enzyme that's essential is completely inactivated at that particular concentration, even though you're more tolerant maybe below that threshold, you'll never get above that because of that. So does your model exclude that relatively simple sort of picture of this?
We're not reaching that yet. We're still at the tolerance strain is 6 grams, and even at that low level, it then produce more.
But why would you think that it would produce more than 20 if it can't grow?
Yeah. No, I don't think so, but other people do. I don't. Yeah.
I mean, it just doesn't make any sense.
No. Yeah. I don't think so. That's right.
So So you also mentioned that you thought methylbutanol might be hard to make because it is so toxic. Have
Yes.
you tried inoculating your cells at higher OD so that they don't need to grow? And see, I mean, can you get a higher, it's a little artificial, I don't know how practical it is, but can you get a much higher growth yield?
Well, that's the funny part of this business is that there's no generality. And so sometimes you inhibit cell growth is good. Other times you inhibit cell growth is not good. Okay, in that case, if you inhibit cell growth, the cell just not do anything. So there's a very complicated physiology there. So if you
viable, if it's something about viability?
No, viability. So that also depends on how we define here. Here, the cell the viability decrease around 5 grams per liter or so, and at 20 grams, they are still active for a certain period of time. But if you use different kind of alcohols, maybe there are other problems. They are not active, even though they are not viable. So the simple definition of tolerance or growth rate or viability is not sufficient to describe all the complex phenomena. Yes?
So are all the enzymes that you put in under constitutive transcriptional control, is there any value in putting the first enzyme of the pathway under an inducible?
These are inducible.
It's all inducible?
It's all inducible, yes.
I see, so you can trigger
Yeah. Yes.
So this is about the very beginning. So the algae stuff. So how does your yield as a function of CO2 partial pressure?
Oh, we haven't studied that. We use the 5% the CO2 bubbling or the bicarbonate. So we have not studied completely. I think Melis will have better idea to answer your question. No, we haven't done that.
Okay, just because when those productivities are
Yeah.
18,000
Sure.
per year or something, I mean, it seems to me unrealistic given what I know.
Sure. Yeah. I don't know how to. Yeah.
Put it up, all right? I mean.
Yeah. It still need to be studied. Yes.
The strain that you evolved with increased tolerance, how many IS elements were in the parent strain? It seems like an unusual mode of evolution that you just got 25 insertion elements in one step.
This is not just one step. This is serial transfer. So, huh? One step. Yeah.
Basically, insertion elements.
Yeah, I think similar the result have been seen if you introduce a stress to E. coli. So this is a high stress condition. So if you just evolve in a nutritional condition, nutritional limitation, then you don't get so much IS insertions. You get SNPs. Okay? So this is an example of cell response to extreme stress. Whether this is general or not, we don't know. But it's consistent with the literature knowledge that the IS element jump under severe stress.
Copy themselves, or did it start?
Yeah, they copy themselves. Yeah.
Okay, well, thank you again.
Thank you.